Okay, so it turns out we do have plenty of time for the panel discussion, so we look forward to your questions. Maybe I can start with a quick one. And Ed, maybe you could tell us, you know, Dana-Farber has been integrated in the cancer cooperative groups for an awful long time. And Dana-Farber has contributed not only scientifically, but financially, a great degree to the support of the groups. You know, and we're finally seeing now credit within the Cancer Center Core Grant and for the work that is done and the contributions that have been made by the center. But you know, what do you see heading into the next 3 to 5 years that we're facing in the current financial environment and the contribution of the Cancer Center directly to the support of the group?
Yeah, I think there are going to be a lot of challenges, obviously. Because of the perfect storm we're facing, nationally, people are going to begin to feel some of what we're already experiencing in Massachusetts, which is a real downward pressure on clinical reimbursements that are used to subsidize a lot of the kinds of contributions the centers can make. NIH funding was already a cost-sharing proposition, and now it's going to be disproportionate cost-sharing unless we trim back our research activities, which no one's particularly anxious to do. I think we're going to have to look for resources to support this from other sources, and I think that the two sources that where there's still opportunity to do that is one, something we haven't talked quite as much about, is partnering with foundations and advocacy groups that that are supporting clinical trials and looking for more impact from that kind of support. And the second is philanthropy. I'm noticing more and more, since we do a lot of philanthropy, that donors, whether they be groups that participate in walks and runs and bike rides, or whether they be things like the Komen, or whether they're high-end kinds of donors, more and more want to see results in the shorter term. And I think that bringing something that looks more like a network that's actually designed, something like the learning system that was described, is likely to cause those groups to generate higher levels of philanthropy than they would in the past. All that said, the total amount of funds to do this is going to be less, and so supporting something is going to mean supporting something else to a lesser extent, and I think we're all just going to have to get more efficient with using fewer funds.
Yeah, Bob Diazio, Mayo Clinic. I had a question for Lisa, basically on the statistical considerations that need to be given for these biomarkers, and specifically how one looks at the differences of the technical platforms. Again, EGFR is always the great example in terms of the initial problems we got into in terms of the testing that was used. It was a non-genomic platform. It seems to me, particularly as genomics and the genomic platforms improve in terms of the ability to make a specific call in terms of the base change that may be present in an individual SNP, one is able to make a much better characterization than with a fish-based assay, antibody-based assay. I'm not sure how you take that into consideration, but one of the difficulties I think that comes from lumping everything as a biomarker that includes genomic as well as antibody and fish-based tests is that it can be really, it can be confusing of all the different types of platforms used.
This is Bob Comas from ECOG Akron and Drexel. So I'd like to ask for a note, I guess, about the issue. Oh, I'm sorry.
Oh, that's okay, Bob. Go ahead.
So I think you make an excellent point, and we have learned that a biomarker is not a biomarker. You have to be very specific about it, and so, but I would caution that even in sequencing, let's say, that I have seen data where you can take the same sample and even the same raw, output, process it using a different alignment method, using a different variant caller, and you can get some very different looking results. So I think what we have to do is try to make sure that we obtain specimens we can use for things like bridging studies. I think we should not rush to prematurely limit the group of patients who can have access to a targeted agent. And we're going to have to figure this out over time. And even people using the same method in different laboratories, I've been amazed at how often you can get different looking results. Additional factors like how the specimen is handled and processed can make an enormous difference. And so I think that we all need to be more aware of these factors, and I would love to have even some of the patients' advocates be involved in this discussion about how can we make sure that, when your sample is taken, that it's treated appropriately, and that you know something about the lab that it's being sent to and the method that's being used in that lab. And so it's really just sort of a tsunami of data, and it's going to take us a while to sort through this. And I mentioned the example of the HER2 story. I mean, we are still, after 12, 13 years, trying to figure out what is the best way to measure HER2. But it's also important to recognize that there is somewhat of a tension between the goals of getting a new therapy out there and identifying the perfect marker for selecting patients for that therapy. It's sort of a trade-off, because you could say, well, when we developed Herceptin, not we personally, but when Herceptin was developed, they had a research-grade assay, but yet it was good enough to find a patient group where we'd be able to get a signal that the treatment was effective. So I think that is a tension that we see over and over and over again, that we want to get a quick answer to, is this drug worth pursuing? And if it's a good enough drug, it's going to shine through despite a bunch of noise. But as we get further down the line and get this out to patients, then we have to start worrying about the details. So how can we collect the data now to help us do that refinement as we really move it out into the community?
Bob.
Sorry.
Okay.
I saw Monica nod. When Monica nods, I speak. So I want to get into the issue of companion diagnostics again and ask Renaud, because he made the point of trying to coordinate the activities of the clinical grade assay and the companion diagnostic to ultimately get it approved. And, as I mentioned yesterday, I think the companion diagnostic idea was a good idea when it started, but it basically locks into a specific assay when the whole dynamics of understanding mutations and understanding interactions in epigenomics really defies locked into a specific assay. And also, it locks in the payer to a specific relationship between the drug and the assay. And it can inhibit the kinds of clinical trials that we were talking about yesterday, where we are ultimately going to be having the biology drive the clinical trials, particularly mutations. So this balance between having a companion diagnostic that locks you in versus, and costs so much time and effort to coordinate between the clinical assay and the companion diagnostic is really an issue, I think, that really needs to be addressed.
Well, actually, that's a very challenging point. The study I described, the discussion started in 2006, and the study started in 2008. Today we are in 2013, and when the result will be available, and hopefully, if it is positive, the drug will be approved. That will be, I don't know, 2015 or 17, and the field will have changed radically. There will be much better technologies available, like deep sequencing will become affordable. So, but still, we have to start at some point when we do a clinical study. And to me, one of the key aspects is to initially have, number one, an informed consent that is written carefully, that allows for the flexibility to not only work with the essay that you have at the start of the study, but to be open to some more exploratory work as better technology becomes available. And it is obviously critical in connection with that to have the ability to store samples for future testing. For example, in the case of FLT3 testing, we will have to do also some comparison with the PCR-based assay with a sequencing assay to see how the two match. That's part of the development efforts. But I think it is very difficult because doing large studies takes a lot of time and technology moves much faster.
I think I just want to add, we've had lots of discussion within the groups about handling the biorepositories and handling the long-term storage and then secondary use of the specimens. And clearly this issue is going to be very important in terms of preserving the capabilities of the bank to do these validation assays when we need to in the future. So we're going to have to take that into account as we decide what really is secondary use of the bank specimens and what we hold back in order to make sure we have the capability to validate when we need to.
Rich Shilsky, ASCO. My question's for Pat. So first of all, thank you for sharing your very inspiring story. It's stories like yours that make us all want to get up and keep going to work every day. But a very important feature of your story is that your oncologist recommended to you your consideration of a clinical trial. And we know from years of experience and even some data that the physician recommendation to a patient to consider a clinical trial is one of the most influential factors in whether or not patients enroll in clinical trials. We also know that unfortunately, excuse me, only a minority of physicians actually are actively engaged in the clinical trials process in oncology. And Bob Comas and I looked at data over many years in the cooperative groups, and I'm sure that the same is still likely to be true today, that the majority of patients who are enrolled in oncology clinical trials are enrolled by a minority of the oncologists who claim to be involved in the process. So I guess my question to you is, when you talk to patients, and advise them as to, what they need to do to optimize their care, how do you discuss this issue with them about, how can they engage their oncologist to ensure that they get a good discussion about a clinical trial opportunity?
Well, first of all, I have to stress that in my experience, my oncologist recommended a clinical trial to me 100% of the time. But you're absolutely right. We see those numbers that the oncologists frequently don't recommend a clinical trial. So when we're out talking to patients, and that's one of the key things that our group back in Grand Rapids talks about, is when we're talking to patients, we encourage them to talk to their oncologists about the options for a clinical trial as possible treatment for their cancer. We aren't out there recommending specific clinical trials. We aren't recommending clinical trials as the end-all, be-all, but we feel very strongly that they should be part of the discussion. And so do your homework. We talk about websites. We give them access to the legitimate websites. We always talk about cancer.gov, clinicaltrials.org, our local GERCOP website, but the ultimate decision goes back to being between the patient and their oncologist, and if the oncologist doesn't recommend it, the patient's not going to participate.
John Mendelson from MD Anderson. I want to go back to the companion diagnostic question. And obviously, when Novartis set up this trial 6 years ago, they had to have a diagnostic in order to do the trial. And that's very, very relevant. But I think we have an unusual situation today. We have Dr. Eichler here from the European Medicines Agency, and we have, sitting in the front row, Dr. Pazdur from our FDA, and we have Lisa McShane here from the NCI, all of whom have thought about this a lot. And many of us feel, and I think it was expressed by Bob Comas, that in 2015 or 16, when your drug hopefully is approved, there will be a variety of assays that might be better than something you designed 6 years ago. And how is the regulatory, what's the advice the regulatory body would give to the world of how to vet and approve a diagnostic assay, forgetting about the fact that when it was all started 6 years ago, it was very important to have it. But today, it might be an Illumina machine or a life technology machine that does it rather than something that didn't even exist then. And is it important to lock a single assay in with a single drug anymore? And Rick, I hope you'll join in on that. I see you nodding your head. Maybe it's all settled and I don't know it.
Well, first of all, you know, I work in CDER, and the in vitro diagnostics are obviously regulated in CDRH, so I cannot speak for them, but I will give you some high-level thoughts because I have thought about these, and I will preface that this represents my own personal opinion. I think, you know, CDER has thought about this, okay? And here again, when we say an FDA-approved test, that's in the label. That's usually in the diagnostic label, the actual specification of those tests, and the indication just reads out as a FDA-approved test. And inherent in that is the knowledge and the belief that these tests will change over time, and that's why there will be bridging studies that are done comparing one in vitro diagnostic to another in vitro diagnostic. But I don't think anyone in CDRH believes that this is a static field and that things in testing these will not change. So this whole concept of bridging studies from one in vitro diagnostic to another is inherent in their philosophy, and this has been discussed extensively with sponsors that really needs to be addressed and needs to be planned out as what the field marches forward in the technical improvement of these tests. So that has been thought about. Now, there are issues that I think we're even encountering now, where you may have multiple sponsors developing multiple similar drugs of a similar class, and they may also be developing different in vitro diagnostics. For example, if somebody is developing a drug against EGFR, they may have different in vitro diagnostics that may measure different mutations. That does pose a practice of medicine situation, because the pathologist is going to go out there, do the in vitro diagnostic that he feels like doing, but that may not be the in vitro diagnostic that was tied to that test. And I think that that's something that the agency and CDRH is really going to have to address, is how do you really ensure that that test matches that drug, or really does it make any difference? And I see that coming out, and that's going to be an emerging problem that we're going to have to address. But the changing platforms and the changing technologies, we've seen that if one takes a look even at the granddaddy of all in vitro diagnostics, ER and PR testing, you know, that has evolved since the mid-1970s, and that has been accomplished through varying bridging studies that have looked at and comparing different technologies. The Herceptin example is a good example of how the test has changed and bridging has occurred through variations in the test procedures.
Yes, but Rick, if I'm not wrong, there are still payers that when you check for RAS, you've got to use the paired assay, which we all know misses some mutations in RAS if you want to get paid for giving the drug. So your philosophy hasn't hit the practice of medicine yet, unfortunately.
I agree with everything Rick said, but I would add to that, and I'm not sure I'm making it easier or more difficult. This isn't an entirely new situation. We'll know that while you develop a drug and you start a long-term study in year 0, life moves on and goalposts may change. And the other situation, for example, that we had recently, there was a drug, placebo-controlled study. In the meantime, another drug was authorized that looked much better in the same indication than the one that was about to come out. Should you license that, knowing that you license an inferior drug, etc.? Sometimes you know that a surrogate endpoint that looked good 5 years ago no longer looks that good. So we have to deal with that situations, I think, in the regulatory environment, not on a day-to-day basis, but it's not entirely new. And I think what you do in that case, you try to exercise some flexibility, and you call it bridging studies. I will call it do some additional post-marketing commitments. So that's the way you sort of have to get around to that.
But do you think those post-marketing studies need to be randomized trials done by cooperative groups, or can something like the CLIA certification group compare, or should the FDA ask for comparisons? What is the test that would be the cheapest and most efficient way to get us past this problem, and yet do the right thing for the patient and for the goal of accuracy?
I don't think there is one size that will fit all situations.
Yeah, I mean, I think it would be impractical to expect we could do randomized trials to address all these things. You know, the issue of getting the concordance between different versions of an assay is not that difficult. You just need the right specimens to be able to do it, and hopefully some basic data about the patients, things like stage.
But who does it and pays for it then?
Right, well, that is the big question. And not only who does the actual concordance, but who's going to pay to have those specimens collected and stored somewhere until we're ready to do our concordance study. You know, what you have to take a bigger leap of faith on, I think, is that suppose you compare two assays and you find a 20% discordance rate. Well, you know, if that 20% is the patients who were sort of only marginally benefiting from the therapy, then you're less concerned. If it's if it's 20% that were really, you know, home run responders to a drug, then it matters. So that's where it gets difficult, I think, to do retrospectively.
Nancy.
Nancy Roach, for colorectal cancer, and I have a comment and then kind of a question. My comment, I was thinking about the unique role of the NCTN, and all this cool science is great, but there's boring clinical trials that can have a real impact on patients' lives. So, for example, there's a trial going on now in colorectal cancer that is looking, it's really boring, but if it works, patients won't end up with neuropathy. And I think, you know, let's not throw out the boring but high impact studies and do only the really cool science. So I just wanted to mention that. The other thing is this all comes back to money. And we're talking about public money. And so we have NCI here, we have FDA here. We have to get the payers to the table. We have to bring CMS to the table to make sure that we can have a real learning system in clinical care. And if oncologists got reimbursed for talking to their patients about a clinical trial, I think that might change things. So maybe the groups and NCI and FDA can force a discussion, or well, you can't force, but prompt a discussion for a pilot project or something like that to see if things like that would actually change accrual, and if research assays would be paid for. And so are there any thoughts about how to make that happen?
So the only one thing I can comment, Nancy, is, you know, some of the discussion yesterday was about the foray of the groups into cancer care delivery, you know, and that arm. And, you know, we've got a very complex environment here. You know, you can just see sitting at the table, and here we're only really talking about more treatment, you know, treatment development today. You know, yesterday you heard more discussion about the broad scope that the traditional the DCP funds through the CCOP Research Base Awards. I think we're all committed to doing that kind of research in parallel with what we do in treatment, because that's the only way we can think of that we can make the bridges that will really keep us to our core mission. And our core mission is to change patient care, you know, to make patient care better and to change impact for the patient. It's frankly not to develop new drugs and not to do biomarker research. It's to change patient care. That's first, second, and third. And you're right, if we're going to do that with the highest impact, because we are the only big leverage system that has its tentacles into every place to make that happen, we are going to have to take that on. And we're going to have to acquire the research expertise and the funding and the capability to really be in that world and not just dabble. because if we dabble, we'll just be wasting resources. So you're right, it's a really big challenge. I think we need to take it on, and we're committed to take it on. But there'll be some very serious discussions for, for the foreseeable future as to how we can really do that best. Jeff.
Yeah, Jeff Abrams, NCI. Two quick comments. I think Nancy might be onto something, because Dr. Kaplan, when he presented the UK system yesterday, showed that in a relatively short amount of time, 10 years, they changed an entire culture amongst the practitioners there by incentivizing participation in clinical trials with financial reimbursement. So I'm not sure that can happen so quickly here in the US. We have a different system. But if patient advocates were looking for a way to truly increase the uptake in clinical trials, that would be a cause certainly worth fighting for. Second, in regards to the companion diagnostics and the bridging studies, I mean, that is one area where the NCTN and the cooperative groups have a real role to play. I look at the Oncotype test, which is widely used now to choose therapy for adjuvant breast cancer. This study came about because of cooperative group studies, the test rather. And the big advantage of doing these big studies is not so much for finding out exactly how to use Oncotype. The big advantage is there's a tumor bank now that won't require us to go and redo the study every time you want to bring a new diagnostic in. But this will be access, public access of all the pharma and diagnostic companies who want to use this to test rapidly hopefully better diagnostics in the future. So I think that's one very important role that the cooperative group program does play. My question was really for Dr. Capville. I really liked your presentation and how you went through the collaboration and broke it down. But I'm wondering, now that you've had the experience in Novartis and looking at the types of studies that we'd all like to do in the future, I would imagine that more of these international collaborative studies are going to be necessary. So what lessons have you learned from this approach, and what would you do things differently in the future? Did this model work well for you?
Well, I mean, that could be a long answer, but I mean, the key is to keep things as simple as possible. I mean, in that particular example, that was a very complex organization. And I mean, it worked. The study enrolled very well, and now we're waiting for the result. But there must be a simpler way to do it. That probably takes a little more time at the beginning to align different people, different groups. But that's worth taking that time. So to me, that's the key.
I just want to add in a comment that speaks to that a little bit, but also everything that's been said this morning. I think we need flexibility in the system. One of our concerns, and I know it's everyone shares this concern, is that as we need to be more and more focused and more of our work is subject to review by the steering committees and by a broad research community, that we lose some of the flexibility that we may need to do some partnerships and some innovative stuff. I mean, it's inevitable. If you're going to get really focused, you lose and straight, you lose flexibility. So finding that balance where the groups can really develop these relationships, which happen, frankly, and have to happen a long time before they hit a steering committee, and to be able to have perhaps the idea is, just to make sure that the entire community, including the steering community, community, are educated into the value of these kinds of resources, of relationships, so that we don't get derailed very late in the process. We need to be very sensitive to that. Rich.
Rich Shilsky. So just one follow-up comment to Nancy Roach's point. And Charlotte will correct me if I'm misremembering this, but one of the deeply buried recommendations in the IOM report that has prompted this meeting now several years later is that the AMA create a CPT code that would improve the reimbursement of physicians for managing patients on clinical trials, acknowledging the greater level of effort that's required to manage a patient who's on a clinical trial as opposed to routine clinical management. That is a recommendation among several recommendations in that report in various domains that have been largely neglected by all the stakeholders and that I think might be revisited.
Yeah, Rich, thanks for bringing that up because you remind me, and again, Cheryl may correct whether this ever made it into a recommendation, but in discussions that we had some years ago in the oncology workforce, we talked about first responders, and this goes to the fact that the majority of patients encounter their first cancer diagnosis and first recommendation for care outside of the centers that are organized more around the clinical research. And so you have to be sure in that setting that the pathologist, the surgeon who takes the tissue, the pathologist who handles it, that all that is happening in a way in this stage of companion diagnostics that the tissues are properly prepared, that the assays, the starting material for these assays, whatever the assays happen to be, are good, that additional imaging or interventional radiology procedures may need to be done, and that right now the mechanisms for funding that in a community setting are almost non-existent unless they have some kind of a formal collaborative relationship with a big cancer center, which immediately shrinks down the number. So that something needs to be built into the reimbursement system. And I think Nancy asked if anybody's tried to do things like this, and there have been, I know of 3 spectacular failures. One, language was in the Kennedy-Hutchison bill. That was going to be the new Cancer Omnibus Act, but that never happened. That bill never happened, in part because of Senator Kennedy's death, in part because it was felt it would be folded into the Affordable Care Act, the larger Health Care Act. It disappeared from that, and we failed in Massachusetts to get that language in, usually on the basis of lobbying from the payer community that this would add to the cost of health care. If you actually look overall for cancer trials, at least, which I think would account for disproportionate amount of this, it adds less than $1 a month to the premium. And I think it's still something that needs to be in there to provide not just the incentive, but the capability that a physician recommending a trial knows that the pathologist is supported, the interventional radiologist is supported, the surgeon is supported, so that all the materials necessary for a 21st century strong clinical trial can be gotten from places that right now would represent maybe 85% of the patients.
So Monica, just to follow Rich's point, which is absolutely correct, that is in the report, but it goes beyond that. You not only have to have a CPT code developed, but it also has to be recognized by the payer. So there's 2 parts of this thing to get it completely done. And the other part of that was we also put in there that you would also have a special CPT code for just offering a trial, because sometimes actually offering the trials and reviewing them, even if the patient doesn't go on it, is a tremendous time spent by the clinicians and their patient together. So that's all in that report. I think you're right. It's buried and we really haven't talked about it very much.
Yeah, the other aspect of this, our interface with the community provider is the one that Robin Zahn brought up yesterday, which is about the electronic health record and the ability to gather research data. It dovetails with the electronic health record and how we have to be really careful as we design these new systems going forward that the clinical researcher is at the table, just getting the clinical researcher at the table when those new systems come on board so that we don't miss an opportunity to again leverage something that we all know is coming that can hopefully be tweaked in a way that will benefit gathering research level data throughout the community. So I think that will also be important. Okay, well, let's take a break for 15 minutes and then we'll go on with the final session of the workshop. Thank you.